Good morning, Satish sir. Thank you for accepting our invitation, sir. Sir, am I audible to you?
Yes, ma'am. Hello. Yes.
Okay. Satish sir, you're not audible.
Hello.
Our expert has joined. I think sir is not audible. Satish sir, I think you need to unmute yourself, sir. Satish sir, shall we start the session?
Yes, ma'am.
Yes, ma'am.
Okay. Okay. So, Pratik, you may start the session.
Should we start now?
Yes, Pratik, please proceed.
Okay, thank you. So now, first of all, uh, a very good morning to all of you for being here, and welcome to this bold new age of cure, where science unlocks what was not secure, from targeted cells to precision's art. Humane the body and uplift the heart, a revolution brewing in every pill to answer mankind's most enduring will. I, Pratik, again welcoming all of you on behalf of Innovative Society. And now, here it is our pleasure to welcome you to day two, all our participants, guests, and organizing team for a 2 day international conference on pharmaceutical industry revolution, a focus on innovation and research, which is organized by Academy Innova World in collaboration with HR Patel Institute of Pharmaceutical Education and Research, Sherpur, and ISTEM Academy. Academy Innova World, it is a hub for research-driven innovation, technical excellence, and the creation of intellectual property IPR. Founded in 2017 as a YouTube channel for aerospace enthusiasts, it now holds as MSME registered STEM research center recognized across India. Unlike conventional academies, our academy operates a dedicated R&D cell collaborating with researchers, scientists, and industry mentors and other candidates, a rare offering in the education sector. Recently, Academy Innova World has been declared as India's first private research and innovation hub, which was featured by Republic News and Gujarat Journal also. Academy Innova World is happy to collaborate with a renowned institution like HR Piper to accelerate innovation, foster crucial collaborations, and translate groundbreaking research into real world therapies that improve global health outcomes. The pharmaceutical industry is revolutionizing through biotech, artificial intelligence, and personalized medicine, aided by regulatory reforms. The International Conference on Pharmaceutical Industry Revolution united scientists, researchers, policymakers, and healthcare experts to explore innovation in drug discovery, manufacturing, and patient care, shaping a transformative future for medicine. I extend my warmest welcome to our esteemed dignitaries, Dr. Aswarya Dhar, founder of Academy Innova World, whose visionary leadership has been instrumental in driving innovation and excellence in research and education. Engineer Pulak Kumar Dhar, Chief Technology Officer and Organizing Chair. He also served as Assistant Scientist at Department of Atomic Energy, whose expertise and dedication continue to guide our technological advancements. I welcome Dr. Sanjay Kumar B. Barvi, Principal HR Piper, Event Coordinator, Mr. Narendra B. Patil, and Mr. Shoaib M. Maniar, HR Piper. I welcome our keynote speaker of the day, Dr. Satish Kumar Sarankar, Professor and Principal, Faculty of Pharmacy, Mansarovar Global University. It's also my pleasure to introduce our keynote speaker of the day, Dr. Satish Kumar Sarankar. He is a dedicated teacher, researcher, and academy leader with more than 16 years of experience in the field of pharmacy. He is currently working as a professor and principal at Faculty of Pharmacy, Mansarovar Global University, Sehar. He also has guided 17 M.Pharma students. One research scholar has been awarded as PhD under his supervision, and currently he is guiding 8 PhD scholars. A gold medalist in Masters in Pharmacy, Dr. Sarankar received the prestigious Inspire Fellowship from the Government of India. He holds 5 patents, has written 3 books, and contributed to several chapters, publishing over 40 research and review papers in reputed journals. His area of interest includes medicinal chemistry, QSAR, in silico drug design, and natural products. He has delivered expert talks at various conferences and webinars and is a member of several editorial and advisory boards. Dr. Sarankar is passionate about teaching, research, and motivating young minds to achieve success in their career. Now, over to you, sir. Please come and take the stage and enlighten us for today's session. Mr. Sarankar, sir. Dr. Sarankar, sir. I'm so sorry.
Thank you, Mr. Prateek. And good morning to everybody. Good morning, all who have joined this conference online. And first of all, I would like to extend my thanks to Dr. Aswaridhar for inviting me to be a keynote speaker in this conference, and everybody who have joined from their institutions, academicians, and industry persons, and all. I welcome you all and thanks the organizer. Today, as per the theme of this conference, I would like to express that how the medicines those are there in the market start from the academic institutions. So while thinking about that, what should I talk in this conference, I decided that I would talk to the about the translational research. So please give me a minute while I share my screen. Is it visible to everybody?
Yes, sir.
Sir,
Yes,
it's
sir.
visible. Can you share the full screen, sir?
Yes. Now, is it okay?
I think, sir, you need to stop sharing and again share it. After presentation mode, again you have to share it, sir.
Okay, okay, okay.
Hmm.
Now, is it visible on full screen mode?
Yes, sir. It's visible, sir. Yeah.
Okay, thank you. So, I chose this topic translational research because means what I thought that whatever the research is conducted at the academic institutions by the budding pharmacists and the researchers in their MPharm students, MPharm scholars who do the research work in their final year, and the PhD scholars also. So those research work should reach to the normal people as the medicine or as the treatment. It should be available to everybody. But what happens that I think that there is a gap there. That gap should be bridged between the industry and the academia, because any idea is started from the academic institute, but to reach to the industry and to reach to the market, that means if we see the history, so there is very less number of available medicines those are marketed, which were started from the academic institutions. Therefore, I thought that yes, we should talk about this collaboration of industry and academia, so that we can give something. Means those research which are conducted in the academic institutes, their results, their compounds, their means whatever synthetic compounds or the plant product or the material, whatever the researchers do. So, therefore. It is important to discuss about this translational research and how this translational research can bridge the academian industry for the next generation development of drugs. So let me talk first about what is translational research. So this translational research is often means we can see that it is bench to beside a bedside approach. Bench to bedside, why we are saying means it is starting from the laboratory in any academic institution, and it is going to the patient. So this is the concept that bench to the bedside approach means it is a process through which we can say that the basic science and insights are systematically transformed into the clinical interventions, and later the final drug is being produced, and it is going to the patient, and it is means reaching to the patient. This particular translational research is a multidisciplinary concept where like I can give the example like molecular biologists are there which can identify the pathways, and some medicinal chemist persons are there who can design the molecules, or pharmacologists are there who test them, and yes, also clinicians are there who provide the insights into the patient need. So collaboratively or multidisciplinarily, this approach can be started together with aim to develop a final compound. So this is generally seen everywhere. Okay, and if we collaborate with the industry, like I am from also an academic institute, so this academic institute when collaborate with the industrial partners, so definitely in the next generations or next upcoming years, something can be achieved very easily, and the cycle or the duration of the discovery can be minimized. Like generally we know that yes, it takes 12 to 15 years to develop a compound, and during those that particular period also, number of molecule, I can say that yes, among lakhs or 10 lakhs of compound, even a single molecules reach to the market. So what about those all lakhs of compounds which were started in the academic institutions, but they could never reach to the industry or to the patient? So, why this translational research is important? This is because if from the starting, if we can take this aim, or if we aim that yes, I want to design this particular compound and it should reach to the patient. So number of things are there which miss at the academic level, because at the academia or any institute, it has its own advantage, and some of the lackings are there which they lack. And if we talk about the industry also, so industry have their own advantages, own working culture. So I would talk differently about the academics role and the industry role. So means academia, we can see that yes, everything starts from here, like the molecular biology, chemistry, pharmacology, and all these things. But let me give you the example of the technologies which are used nowadays. Like I would say about the CRISPR gene editing technology. This gene editing technology was started uh, many years ago in the academic institution, but now this technology has reached to a place where it is being used everywhere now in the care of the patients. And like in the drug discovery process, in the preclinical validation also, like the drugs or the molecules which are prepared in the labs of the academic institutions are tested in on animal models, and they are validated, and initially they provide the idea that the particular molecule will be active for the management or the treatment of any particular disease or not. So this is also important from academics. Now, I would talk about the talent development. Talent development means what? That yes, academic institutions are there who initiates, who develop the talent in the students or the research scholars, and they prepare them. And I mean, means I can say that yes, academic institutions are like the nurseries for the means these translational people who will now can serve as the scientists or the researchers with a vision to develop a very good molecule that will reach ultimately to the patients. And yes, this foundation sciences, we can say, like ethnopharmacology is there, or the traditional medicinal knowledge is there. So number of things are there. And then, like I should give the example of artemisinin. Artemisinin was means started from the traditional knowledge, and now it is a very means huge level. It is being used for the treatment and management of malaria. It is an anti-malarial agent. So this traditional or ethnopharmacological is also important, and through this academic institutions, all this knowledge is provided to a person, a researcher, or the scholar to take this idea, and later these ideas are converted into the things those are helpful for the people. Now we must talk about the industry role also. Industries are working on their own culture, means they do not start from the means basics. Yes, they may also, but yes, they work on the scaling of those molecules. Means industry is responsible for scaling and innovations. Those academic discoveries are there. So industry transform them into the manufacturable or the stable and commercial viable therapies. And then comes the clinical development. Number of things are there, like the clinical trials, we can say. So industries are responsible and they take care of these clinical trials and regulatory approvals and establishing good manufacturing practices. And without all this, even the means most promising molecule, if any is developed at the academic institute, can never reach to the means patient. Means the curiosity of any academic person remains at the uh, person's level or the academic institutions level without the help of the industry. And finally, the role of industry in its market delivery. This is the very big thing that means most patient-centric role, we can say it, that the distribution channel, global supply chain, and affordable access models are there through which the industry uh, provides the drug or the means molecules to the patients. Everybody has seen this COVID-19 example. So the development of the COVID-19 vaccine is the very good example where we can learn through that. Yes, the concept of the mRNA platforms and viral genome, all these things started in the academic institutions, but later with the partnership of the pharmaceutical companies, those companies manufactured these vaccines very easily within a very short period of time. That means I was talking that yes, 10 to 12 years are taken, but we have all witnessed that yes, within a short period of 1.5 to 2 years, these pharmaceutical industries have developed the COVID vaccine and delivered to each and every person of India in a record time. So this is the kind of research or the means collaboration I am talking about, that if this such kind of collaboration will be there, so whatever I am means aiming to discuss or to deliver can be achieved within a very short period of time. So why this academia and industry gap is to be bridged means, like I can say that there is a gap or a valley. That valley is to be bridged. Like at one side, if we can say the ideas are there, the molecules are there, compounds are there, and the other side of that valley, that patients who are waiting for good molecules to be available to patients. So that valley can be bridged so that everybody should be benefited. So this bridge is can be built through the public-private partnerships and collaborative pools expertise, data, and resources to accelerate the development. Uh, like we can see that if anybody is sitting anywhere in the world, so through this approach, any drug can be made available to the person everywhere with the help of industries. The industries is also responsible for the technology transfer, like means if we want to bridge this gap, so we can say that technology transfer is the key tool. Like today, universities have many people who are specialized in their areas that handle intellectual property rights also. So there, yes, this IPR, intellectual property rights, I would say that yes, sometimes why there is a gap, why industries do not generally collaborate with the academic institution, because it is also a problem of IPR. Industries want their own, and academic institutions or the academic people want their own IPRs to have. Okay, so there is a problem, but that can be mutually solved through the collaborative approach. So in this way means the discoveries move smoothly, and the academic research will not be limited to the only research paper or any conferences, or it will not stuck somewhere in the pipeline of the drug discovery. And yes, I must say that academic institutions lack the infrastructure or the technologies. So those technology and the infrastructures can be shared with the industries, like the incubators and the translational hubs are there. So with these all, the researcher and the entrepreneurs can work together and this gap can be bridged. And of course, collaborative funding mechanisms can be there, and these through this funding mechanism, jointly, with the government also provide the funds for the research, and industry and academic bodies, if they all together join and do the collaborative research work, so it will reduce the risk also, and it will allow the ideas to flow to the marketplace very easily. So without this, I must say that this without this bridge, discoveries remain disconnected. So anything which is being discovered at the industrial level is not reach to they do not reach to the patients. So means if all this collaborative approach can be uh, joined together, so very easily the things can be achieved. Now, this next generation drug development trends are there that which can help to bridge the gap and to fasten the means to make the drug development process very fast, artificial intelligence is there, digital twins technology is there. So these are not there just to analyze any data or means something, but yes, these artificial intelligence nowadays, machine learning technologies nowadays are designing the molecule, and number of softwares are available, pages are available where we can predict the toxicity, ADME profile of the molecule, and even means we can simulate the entire clinical trial using the digital patients models. So this artificial intelligence is quite important nowadays, and the time being consumed in the actual clinical trial can be reduced by using these digital patient models. Second one is this RNA therapeutics. So I gave you the example already that yes, while COVID-19, RNA vaccine was developed, and means it is a real-time revolution. I can say that yes. And apart from this COVID-19 vaccine, 19 vaccine, RNA-based treatment are there for rare diseases some of the cancers are also cured by this RNA therapeutics, and even personalized medicines are now in development through using this RNA therapeutics. Next thing is this cell and gene therapy. So I can say that yes, we are entering to an era where the cells themselves can become the medicine. It's T cell therapy is there. I was when reading. So this give hope to the patient that yes, this can be useful for the treatment of many kind of cancers. And gene editing is also there that is used in the treatment of number of incurable diseases which were means very tough to cure earlier, and through these technologies they are now easily cured. The concept of precision medicine is there, means yes, we cannot say that yes, one size fits all like concept. By using this genomics and RNA therapeutics, cell technology, gene therapy, so now it is possible to design individual medicine for individual person, or that means we cannot say that yes, one drug is being like in today's era, the one drug is there and it is given to every patient. So through this precision medicine approach, individual patient will get the drug for his requirement or her requirement as required. Now there are some challenges and opportunities. I want to discuss that. What are the challenges? So in this translational research, some regulatory complexes are there. But if we talk about the academic from the academic side, so yeah, it is tough. But with the help of the industrial people or some of the licensing persons, these approvals are can be made very fast, and industries can get the approval easily. Another challenge is high research and development cost. Yes, it is quite high and it takes time also. So by using the latest technologies, like I was talking about, artificial intelligence and this machine learning, number of tools are there, like nowadays computational scientists use, like QSAR is there and docking is there. So the process of developing a compound or a lead is reduced, so the cost will be reduced also and the time will be reduced also. And the data sharing barriers are there, some, like industries easily do not share their data with the academic institutions. So later with the means collaboration, this can be minimized. But yes, if there are challenges, so some of the opportunities are also there, like through the global collaborations, are this like this global collaborations are nowadays very accelerating, like vaccines are being developed. This COVID vaccine, we can take the example. Some adaptive trial designs are there, and digital innovations, from the artificial algorithms to the blockchain in clinical trials, is transforming. So this research, overall, all the research technology, or the means methodology, is being changed, and it is ensuring the transparency also, and it is speeding up the process also of the research. Now, what can be the vision as we look ahead? So, what is the vision for the translational research? So, I see that with the emergence of this translational ecosystems, like network, where the academia and startups are there, pharma companies are there, so they can be interconnected. And if they start exchanging the data and the ideas in real time, so yes, definitely this translational research approach will be benefit for the patient care. And yes, patient centric discovery, where means I should say that yes, every stage of drug development uh, begins not just in keeping mind that yes, I want to design or develop a single molecule, but aim should be there. Yes, this particular molecule should reach to the patient. So if from the starting, at any academician keep in mind that it should reach, so from the early stage, the person should collaborate with everybody, whosoever will be means the stakeholder in the process of delivering that molecule to the patient as the drug. And this rapid translational research can be a very good future in the scientific discovery in the upcoming years. So this was all that I wanted to talk about. Through this translational approach, we can uh, have a molecule that is developed at the institutional level to available to the patients. Now,
Sir, thank you so much, sir, for an wonderful insights. And you have clearly picturized the thing. What are the industrial applications that can be, can be done? Thank you, sir.
Thank you so much, sir, for this informative session. And this session not only told about what this technology is, but also emphasized and focused on the challenges we are having ahead. And for the new game changers, it can be a very helpful insight, so that whenever they will want to pursue their further education and they want to go in this field, they will also be aware of the challenges they will have to face, and they can also just make their minds to face those challenges. So thank you so much for all these insights, sir, and it was very helpful. Now, moving forward with this, I would like to I would like to just inform here, and it's time to begin the presentation for this conference. So I request Mr. Narendra B. Patil, sir, to take over the session as the session chair. Sir, please.
Satish, sir, once
Narendra.
again, thank you, sir, for accepting our invitation, sir.
Thank you, madam, for invitation, and I'm very much thankful to the entire team. So, can I leave now, madam?
Yes, sir. Yes, sir. Thank you, sir. Thank you.
Okay.
Narendra, sir, please take over the session, sir. Yeah. Narendra, sir, am I audible to you, sir?
Yes, ma'am. Audible. Hello.
Yes, sir, you're audible, sir. So we can formally start the presentation, sir.
Yes, ma'am. Hello. I am audible, ma'am.
Yes, sir, you're
Hello.
audible, sir.
Now we can proceed a poster presentation. First poster is Srishti Patil and Krishna Thakur. Display your poster and start your presentation. Hello. Krishna Thakur and hello. Krishna Thakur. Display your presentation. Hello. Thakur Krishna. Hello, ma'am. Wait up 5 minutes. Next group, Yogesh Patil. Display your poster. Hello.
Yes, sir. Yes, sir.
Share your screen and start your presentation. Say, right, poster presentation. Hello.
Yes, sir. Sir, can I start presentation?
Yes, yes, start. Ma'am,
Okay,
shall I start?
so uh, good morning, respected teachers and my dear friends. I am Sujal Shah, a student of TYB Farm from College HR Patel Institute of Pharmaceutical Education and Research. So, my topic for a poster presentation is green solvents, unlocking the potential of natural deep eutectic solvents in pharmaceutical applications. So, it is a very emerging and fascinating topic for pharmaceutical industries. So, in recent years, the pharmaceutical industry has been facing a major challenge: the how to balance efficiency and innovation with sustainable and safety. Okay. The traditional organic solvents, although widely used, are often toxic and volatile and environmentally harmful. This is where the concept of green solvents comes into play, solvents that are eco-friendly, safe, and sustainable. So, among the many green solvent system, one category has gained special attention. That is our topic. That is NETs, natural deep eutectic solvents. So, these are mixtures of natural compounds such as amino acids, sugars, organic acids, and choline derivatives. When we combine a certain ratio, they form a eutectic mixture, which have a melting point less than individual components. So these solvents are biodegradable, non-toxic, inexpensive, and highly tunable compared to organic solvents. So now you may ask the question. Now you may ask the question: why are nets so promising for pharmaceuticals? Firstly, they improve solubility and stability of poorly water-soluble drugs, which makes the medicine more effective and better absorbed in the body. Secondly, they enhance extraction of bioactive compounds from natural products, opening new doors for herbal drug formulations. Third, uh, it helps Third, it helps in the drug delivery system. Nets can act as a carrier for drug delivery systems, providing controlled release and reducing side effects. So these are the applications of the nets compared to organic solvents. These are also can be used as a preservative, solubility enhancement, bioliability enhancement, extraction of bioactive compounds, drug delivery, chromatographic media. So these solvents can be very useful in green chemistry and sustainable industry. These can be also used in various drug delivery systems and can be helps to absorb the drug properly in the body. These are can be also used in the food and nutrient delivery, which helps to absorb the nutrients properly in the body. So these are the various benefits of the using nets instead of organic solvents. Compared to organic solvents, nets be very biodegradable, non-toxic, inexpensive, and highly tunable in the applications. So in today's pharmaceutical landscape, we really need such type of solvents to carry out various process in the industries. Thank you, everyone.
Hello.
Yes, sir.
If any question to ask, please raise your hand and ask one by one.
Okay, I have one question. What's the application of this?
Ma'am, these can be used as a alternative for organic solvents in the industry.
Okay, and already we have some farming happening, right? So how it is different from the existing farming that is already happening?
Ma'am, these solvents will provide various benefits compared to other solvents, such as these are not environmental harmful, non-toxic, and will be very easily biodegradable.
Biotic. Okay, okay, thank you.
Okay. Thank you, ma'am.
Hello, ma'am. Hello. Hello. Hello, ma'am.
Yes, sir.
We can proceed the second poster. Hello. Hello.
Yes, you are audible and we can proceed.
Yes, yes. Now, second poster for Krishna Thakur. Share your screen and start your presentation.
Okay, sir.
Yes.
So, good morning, good afternoon to all the dignitaries of this online conference. I wish my heartful gratitude for providing us the opportunity to share our presentation. My name is Krishna Thakur. I am pursuing my education from third year BPharm at HR Paper. As we know, we sometimes fracture our bones, and the orthopedics suggest us to apply a well-known traditional cast, which is made up from POP or also known as gypsum. But at a point of view, it has several demerits like it has a heavy weight, it reacts with water and cause foul smell, and mostly the casts are irritable to skin, which causes redness of skin due to unbreathable application. So for this, we have working on a project named as Biocast, which can stand as an eco-friendly solution for the traditional way. This biocast uses a filling material known as a liquid resin, which is naturally extracted from the plant materials. This material is naturally extracted and is modified to increase the strength by adding the fibers and nanoparticles like naturally extracted, which are distributed in the liquid mix. Going through the advantages of this, firstly, it is a patient-friendly cast which need a short time for curing and has a good strength. Secondly, it does not cause any irritation to skin and infection to skin because it gives a free breathable feeling. It also releases the water to avoid the absorption of water, unlike in the traditional cast. Speaking of for the other applications, like it has strength as the flex, we can modify it by various flasks like jutes and various types of natural fibers. Um, it requires training requirement very shortly because it can set very at very low period within just 10 to 15 minutes. And Speaking for the conclusion, the biocast can be good and a better replacement for the traditionally used gypsum cast, as it has a better properties than that of the traditional cast. The biocast is breathable and can provide a good airflow that will prevent the redness and irritation of the skin, which may cause discomfort of the patient. Unlike the traditional cast, they do not show any type of participation with water or heat. Sometimes the traditional cast form a foul odor due to water absorption, and this can be avoided in the modern solution as a biocast. So I conclude my presentation and thank you for sharing your valuable time with me. Thank you,
Hello,
sir.
ma'am. If any questions? Hello.
Mhm.
If any question, raise your hand and ask to me.
Hello.
Hello. Any question?
Sir, hello.
Hello. Hello, Krishna.
Yes, sir.
If any questions, sir?
Sir, anyone can ask questions.
Krishna, how to be it is effective bypass than other biomaterial?
Sir, we can use various types of hardeners in it. It can be naturally or chemically derived.
Yeah. Okay.
Mm.
Have any
Thank
question?
you.
Yes, Krishna. Stop yourself.
Okay, sir.
Now, Divya Shah and Nisha Patil, share your screen. Divya Shah, Nisha Patil,
Yes, sir.
share your screen and start your presentation. Divesh, start your presentation.
Good morning, everyone.
Hello, start your presentation. You are audible.
Good morning, everyone. I'm Divesh Shah, and with my co-partner Nisha Patil, we are a third year B Pharma student from the HR Patel Institute, Shirpur. Nowadays, there is an increase in the ratio of multiple severe types of diseases. From these severe types of diseases, we done the research on the main disease, that is urolithiasis, which generally known as the kidney stone. Then, how kidney stone form into the kidney? Generally, the urine consists of the calcium, sodium, potassium, uric acid, and oxalate. If the level of these increases into the urinary tract, then it causes the scratches to the wall and it forms the crystals. And it is a very painful condition, and the urine volume is also decreases. So, on this harmful and severe disease, we prepare the extract from the Acheranthus asparaginum plant, which belong to the Amaranthacea family. So we done its in vivo and in vitro studies, and this is actually effective for the anti-urolithiatic activity. So in this plant, we check the multiple types of constituent like alkaloid, saponin, tannin, carbohydrate. Alkaloid is done by the test, drag drop test, and the saponin by the foam test. In this way, the mainly type of specialized test, we can check it. So first of all, we do the in vitro condition by the two types of assay, nucleation assay and aggregation assay. In the nucleation type of assay, we prepare the buffer solution in which we use the calcium chloride, then oxalate and sodium chloride. So in that way, we take the two types of solution, stock solution and the sample solution. In the stock solution, we use the standard like a cystone and the calcium chloride, oxalate plus water. And in the sample, we use the aspera plant and the calcium chloride and oxalate. And in this way, we form the crystal and you incubate it and form the oxalate in there. And we found the turbidity in it, and we can see that in the cystone, which is our standard, and there is the sample, which is the aspera plant, and it matches to the nucleation assay. And in this way, it is results that our formulation is matches to the standard. And in the aggregation assay, we also take the cystones and extract and matches with the standard. And for this, we also done the in vivo studies or assay. And for in vivo, generally we know it is done in the rat or the mice. So that's why I take here the mice known as Vista rat, and here I prepare the 5 groups, and each group consists of 3 rats. And in this rat, in group number 1, I treated it with the food and water for the 28 days. And in group number 2, I induced the stone orally by the ethylene glycol. And in group number 3, I give them the standard and form the cystone in it. And in group number 4, there we give the extract with the 200 mg per kg. And in group number 5, I increase the level or the quantity of the extract from 200 to 500 mg per kg. And it literally give the effective and anti-urolithiatic activity. So as we see in group number 1, there is the urine volume is 9.2, and if we check the standard, there is the urine volume is 10.2. And in group number 4, in which 200 mg drug is given. Their urine volume is 10.2, and here there is in group number 5, in which I increases the extract volume. There is the 12.5, the 12.3 is the urine volume. So in this way, your for calcium crystals and phosphate crystals are made. It clears the passage of the urinary tract, and their urine volume is increases. We know the pH is always the same, and the uric acid is also decreases, and calcium and phosphorus level is also decreases. So in this way, this is the effective with the standard, and their main confusion is And their main confusion is in vitro study, generally we matches with the cystone, and there it show the anti-urolithiatic activity plus increases the volume of the urine, and it also give the nutriatic activity. And in vivo studies, we we generally melts the calcium oxalate crystals, and it also give the nutriatic activity. So this is my presentation towards the urolithiatic disease, and it is literally effective regarding to kidney stone, which is the major problem nowadays.
Any questions? Hello.
So in this way, I can conclude my topic here. If anyone have any question, please.
Yeah, thank you. It's quite insightful, knowing that how we are moving so fast towards the Ayurveda again and using all those botanical beings to cure that kind of disease. But I mean, as we are also talking about the botanicals, there are terpenes and there are many more molecules there, combinations which we talked earlier. So can you just more about all those like their side effects they might cause or for the supervision which comes up? Can you go ahead and like those things also?
Excuse me, sir. Sorry for interruption, but I'm not properly audible.
Hello, Divya.
Hello.
Hello.
How do you now?
No, sir. Sorry, but it's not clear voice, sir.
Okay, okay, just give me a moment. Okay, am I audible now, Divya? I'm sorry.
Yes, sir.
Okay, thank you. So, Divya, as I mean, that's quite insightful knowing about that scientist Athera and all those cures it can provide us with a vast range of opportunities lying ahead. And it's quite good to know that nowadays youngsters are moving towards the Ayurveda again, opting for the botanicals. And but as we are talking about the botanicals, the constituents are there. So, I mean, can you also enlighten about the side effects or the supervisions we should be using all these drugs before we proceed further? That will be of more help for everyone when they will know also about the side effects because you also mentioned the constituents. So there are some constituents which can cause like the infertility or they might be harmful to breastfeeding. So can you go ahead and enlighten more about that?
Um, Sir, generally it's not. Am I audible, sir?
Yeah, clear. Go ahead.
Sir, generally it. Sir, generally this plant is not produce any type of toxicity, because there is the concentrate. These studies is generally based on the various types of concentration. So if we done these studies on the concentration basis, and we give the proper concentration in this extract. So that way it can't provide any toxicity or infertility like this. There is no harm
Okay.
for this.
Okay, no problem. I mean, you should just go ahead into the topic, and you chose quite insightful was there and the way you presented. So you go ahead and research more about all of it. That will be very helpful for you and upcoming generation, the way you're interested in all that. So thank you very much, Divya.
Yeah.
Over to you, Naren,
Thank
sir.
you so much, sir. Thank you so much, sir.
And sir, Naren, sir,
Hello.
yeah, please proceed further with the presentation, sir.
Yes, sir. Yes. Divya, you can start your share.
Yeah, sir. Yeah.
Hello. Minakshi Sadukia Kairadi Sisodia. Share your screen and start your presentation.
Yes, sir.
Good afternoon, respected dignitaries. Myself, Kairavi Sisodia, a student of HR Piper Pharmacy College and a student of TYB Farm. My co-partner is Meenakshi Salunke. Today I am presenting this topic Ayurveda Ayurvedic cough syrup. We know that Ayurveda in our tradition so using Indian traditional system and we prepare Ayurveda cough syrup. Then,
Hello. Kairavi.
sir, I am audible.
Audible. Share your screen.
Sir.
Firstly, share your screen and then start with your presentation.
Sir, 5 minutes wait. Sorry, sir. Sir, I'm audible.
Yes, yes. You can start your presentation.
Good afternoon, respected dignitaries. Myself, Kairavi Sisodia, student of HR Patel Institute of Pharmaceutical Education and Research, a student of TYB Farm. My co-partner is Meenakshi Salunke. Today, I'm presenting this topic is Ayurveda Ayurvedic cough syrup. First of all, we know that Ayurveda in our Indian tradition. So using Indian tradition system we prepare Ayurvedic cough syrup. In this cough syrup, main active ingredients in cow urine and banana stem. We using the formulated Ayurveda containing cow urine and banana stem provide better alternative for the treatment of cough, because the day by day the cough growth rate is increased in 2023 to 2028, because Ayurveda formulation is no side effect and a better therapeutic effect, and both the cow urine and banana stem have potential medicinal properties like the antibacterial, antifungal, and anti-inflammatory, and those the expectorant properties. A traditional method using both the cough syrups, banana stem, and cow urine, uh, using extract of natural expectorant like banana stem extract helps in lose the mucus, help secretion, expel, and urine enhance the respiratory function. And the antimicrobial properties, those the cow urine possess the activity shows the and associated with the cough syrup, and both the anti-inflammatory and rich in nutrients. We should prepare we Ayurveda and Ayurvedic preparation. Ayurvedic preparation is a no side effect and no harmful effect. Shows the as compared to the allopathic cough syrups. And then result and discussion. First of all, appearance is cow urine is pale yellow and the odor bitters and musky. Material ingredients like the stem, tulsi, glycerin, rose water, ashwagandha, use in make the and ensure safety and efficacy of the product. On traditional medicine system, we ensure the while spread of easy access to the formulation and provide like events. Why should we prepare in Ayurveda? Well, first of all, marketed and Ayurveda. First of all, marketed preparation is very costly. Ayurveda preparation is a cost effective. And then use of preservative. Ayurveda is no preservative. Because the marketed preparation is synthetic and Ayurveda is natural, because no side effect and no harmful effect, and marketed preparation is a harmful preparation. Sir, I am audible.
Hello. Any question ask any query.
Sir, final conclusion is Ayurveda was developed in the cow urine and for is extract in therapeutic applications and by utilizing cost effective and natural resources. Thank you, sir.
Whether I
Any
am audible?
questions? Yes, sir.
Ayurveda is how to be masking the taste as compared to the organic preparation or marketed preparation will be effective.
Sir, we in Ayurveda preparation we are using only
Hello.
in and the banana stem.
No. Bhairavi? Hello.
Yes, sir. Yes, sir.
Sir, any question? Hello. Pratik sir, any question? Hello. Pratik sir. Any question, sir?
No.
We can proceed next. Okay. Bhairavi, remove. Stop your presentation. Stop your screen.
Yes, sir.
Next, Thakre Divya.
Yes, sir.
Share your screen and start your presentation.
Can I start, sir? Hello, sir.
Start your presentation.
Okay. So good afternoon to all. I am Divya Thakre, third year student of HR Piper. I am here to present my poster on topic Innovative Herbal Approaches to Combat Lumpy Skin Disease in Cattle, a systematic approach. Lumpy skin disease, commonly known as LSD, is caused by the Capripox virus of Poxviridae family. It is an emerging infectious disease of cattle that possesses a major threat to livestock health and dairy economy, particularly in India. The disease is spread mainly through the insect vector such as mosquitoes, flies, and ticks, but also spread by the direct contact through the saliva, nasal discharge, milk from infected animals. LSD is a pandemic mainly in Africa countries. The clinical signs include nodules, skin, high fever, reduced milk production. In severe cases, may lead to abortion and even death of animal also. Conventional treatment mainly relies on vaccination and supportive therapies. However, they are not always accessible, affordable, or fully effective. So, the herbal remedies can provide an effective, economical, and eco-friendly alternative. So, my poster highlighted a herbal spray formulation or oral herbal formulation spray using natural ingredients. So first of all, first is herbal spray formulation. Herbal spray formulation prepared by using a natural ingredient like neem leaves extract, which have antibacterial activity. Then tulsi leaves extract, which have antiviral activity. Orange peel extract, which have anti-inflammatory activity. Banana peel extract, which have antioxidant activity. Turmeric extract, tea tree oil extract, eucalyptus oil, coconut oil. All these are used in the formulation of herbal spray formulation. Then next is the oral herbal formulation prepared by using bay leaves, tulsi leaves, garlic, banana peel, turmeric, betel leaves, neem leaves, amla, orange peel, ginger, cumin seeds, jejri. This also have shows the different activities. For example, antioxidant, antiviral, etc. This formulation can be used as a dietary supplement to improve digestion, immunity, and overall health of cattle. The ingredients were carefully processed into the powder, then extract it, ensuring the standard quality and easy application. It is very easy process to apply on the animals.
The main conclusion of this study is that herbal remedies can reduce the severity of symptoms, support recovery, and minimize the economic losses due to the LSD. They are safe, affordable, sustainable compared to the conventional drugs. So, this is the main conclusion of this project. Thank you, sir.
Thank you, Divya. If any question, ask to the Divya. Hello, Pratik sir. Hello. Divya. Hello.
Yes, sir.
This formulation, how to we characterize, or in which
Sorry,
mechanism?
sir.
Hello.
Yes,
I'm
sir.
audible. This
Yes,
formulation
sir.
in which mechanism will we act on the lumpy disease?
So when we give these drugs to the animal, they affect as the anti-inflammatory or antiviral in the animals.
And how to we characterize this formulation? In which parameter you have characterized?
Sorry, sir.
Which parameter you have? Hello.
Yes, sir.
Which parameter you have performed to optimize this formulation?
Sir, we prepared the solution of neem leaf extract, tulsi leaf extract, banana peel extract, because they have the different activities like antibacterial, antiviral.
Okay. Thank you.
Okay, sir.
Next. Neeraj Pardesh. Hello.
Yes, sir.
Share your screen and start the presentation.
Yes, sir. Sorry. Sir, may I audible to all?
Yes. Start.
Okay. So, good afternoon, everyone. Myself, Neeraj Siya Deepak Siya Pardeshi, the final year student of HR Patel Institute of Pharmaceutical Education and Research, Shirpur. So, my topic is sustained bioremediation of the chromium 6 adsorption from the pollutant water via modified plant, ginger and algal spirulina biosorbent. So, as we all know, heavy metals are the toxic pollutants and which are non-biodegradable, which are present in the air, soil, and water. The analysis from the Central Pollution Control Board, which is located in the United States board, so they conclude that the India have the 2 mg per liter of chromium hexavalent in water. Generally, 26 to 25 ppm concentration of the chromium is found in the small aquatic bodies, and also in the soil it is found in the 1 to 3000 ppm in the soil. So as we all know, the heavy metals are the toxic and cause the various effects. So this study include the chromium hexavalent majorly because in aqueous form, the chromium is present in the two form. One is a trivalent chromium and another is a hexavalent chromium. From that, hexavalent chromium is a more reactive, and so that it can cause the neurological, reproductive, respiratory, and some metabolic disorders. Also, it can generate the free radicals and act as a carcinogens in the lungs also. So when the tanneries industries or electroplating industries release their water into the small lakes or rivers, the chromium hexavalents are also released into the water, and they can cause some disorders like neurological and reproductive also. So there are some conventional methods and new technologies also. In conventional technology, we use some sea species, bacteria, fungal also use, but they can form the secondary sewage into the water, so that we avoid them. And some new technologies like reverse osmosis and filtration and chemical treatment are too expensive. To overcome such a problems, we use the spirulina and ginger. As we all know, spirulina contains the carbohydrates, proteins, fatty acid, minerals, and main therapeutic protein is a phycocyanobate. Also, ginger contain the more than 20 phenolic phytochemicals, which includes the uh, gingerols and dihedral terpenoids, which is a pre-oxidant and have ability to bind with the metal. So it can help to reduce the metal concentration and reduce the oxidative stress in aquatic life, which is caused by the chromium hexavalent. So this study includes the use of the spirulina and ginger. So first we see the chemical modification of the spirulina and ginger. So we take this ginger and spirulina, wash them properly and dried for the 20 days in the sunlight. After that, grinding is performed, and by the using of sieves, the uniform particle size are used. After that, for the chemical modification, the sieve powders we taken and treated with the zero point 0.1 normal NaOH, and this slurry is mixed at the 300 rpm with the using of magnetic stirrer for the 1 hour at room temperature. This process helps to remove the additional alkali from that. Then powder is dried and sieve. Then wash with the distilled water twice. Then addition of citric acid is done. Then after the drying, again washing is occur with the help of Whatman filter paper. After that, the biomass is dried overnight at 50 degrees Celsius and stored in the airtight container. This process helps to introduce the anionic surface groups such as the hydroxy and carboxylic to enhance the uptake of the chromium hexavalent. So this analysis done by the FTR technique, as mentioned in the poster also. So FTR technique used, where that first we analyze the natural spectra of ginger and spirulina, so that we can conclude that the touching at the 3431 centimeter inverse, we found the stretching vibration that include the carboxylic, NH and phenolic group. After the chemical modification, uh, we can find the peak at the 3362 centimeter inverse, that help to indicate the successfully introduction of the functionalizing group like carboxylic and hydroxy group. Then various studies are done related to the pH also and temperature also. These kinetic based studies we perform by preparing the stock solution of chromium hexavalent, which is made up at the lab scale by using the potassium dichromate, and the ppm solution is prepared with the help of the mixing with phosphoric acid, which help to adjust the pH, and diphenyl carbazamide reagent. So this stock solution is analyzed by using various kinetic batches, like effect of pH, effect of adsorbance of dosage form, then effect of contact time, and effect of temperature on the biomedical absorption of the chromium hexavalent. So first we conclude the effect of pH. We take the various pH ranges from starting from the 1 to 8. In that we mix the 1 gram of biosorbent with the 50 mg per liter of prepared stock solution of chromium. So in that from the graph we can conclude that the result is optimum obtained at the pH above than the 6. As we increase the pH, the hydrogen common ion effect also increases. In the lower pH, the concentration of H plus proton also increases, so that they can interpret it with the binding of the biosorbent with the metal ions, so that we increase the pH, and which are help to absorb the metal ions. another is effect of contact time. We take the 10-minute up to 90-minute time. In that effective time is observed that 80-minute it is a optimum time to contact with biosorbent in the that particular solution. After that, one bioassay also studied, in which we take the zebrafish, and two groups are divided. Likewise, one is a control and another is a text group, in which we use water tanks. In control group and test group, we put the prepared stock solution of chromium hexavalent, and after that, by using following the various protocols, we use their levers to detect the hepatotoxicity due to the chromium in a zebrafish. So we centrifuge we homogenate and centrifuge the process, and the pellets are collected and analyzed. So as we mentioned in the poster, so the control group having the more hepatotoxicity, then natural and less toxicity is seen into the chemically modified ginger and spirulina. So overall conclusion is of this study is to adsorption property of spirulina and ginger. Also, they don't have any side effect, because the ginger and spirulina also act as a food for the aquatic bodies, which not form the secondary slug into the environment and not any harmful. Other than they have some properties like anti-inflammatory and antimicrobial properties also. So that this study helped to adsorb the chromium ion, which is present in the most of the hexavalent form in the industrial or territorial industrial water. So if you have any question, you can ask, and thank you for the institute for giving such opportunity. If you
Thank
have any question, you can ask.
thank you, Neeraj. Any question? Any question, ma'am? Aishwarya, ma'am? Hello? Okay, Neeraj, no question. Neeraj, stop yourself. Mukti Dasputi, share your screen and start your presentation. Dasputi, hello. Dasputi Trupti,
Yes, sir.
I'm audible.
Am I audible?
Yes, audible. Start your screen.
Good afternoon, sir. Good afternoon, ma'am. My name is Divya Chandra Singh Girase with my colleague Trupti Mahindra Dashpute, student of third year B Farm at HR Patel Institute of Pharmaceutical Education and Research, Shirpur. Today we are presenting our work, cow urine based serum with hyaluronic acid and antioxidant, a breakthrough in skin rejuvenation. Our research focus on developing a natural, innovative, and cost-effective anti-aging solution for skincare. As we know, skin aging is a gradual process caused by both intrinsic factors such as genetics and hormones, and intrinsic factors like sun exposure, stress, and pollution. This leads to the wrinkles, fine lines, dryness, and loss of the elasticity. Research shows that over a 2,000 million people worldwide are concerned about aging skin, which highlights the demand for affordable and effective solutions. Therefore, we aim to design a serum that not only delays a visible signs of aging, but also promotes skin rejuvenation using natural and bioactive components. Our aim was to formulate a novel cow urine based serum enriched with hyaluronic acid, collagen, antioxidants, and vitamins to enhance hydration, skin repair, and protection against the environmental damages. We followed a systematic formulation process, which includes preparation of the water phase, oil phase, combination of both of the phases, cooling with collagen, pH adjustment, and addition of the preservatives. Cow urine was used as a natural bioenhancer along with the hyaluronic acid for hydration, vitamin C for the antioxidation, and collagen for elasticity.
Sir, here we studied some parameters. Firstly, we studied the DAC thermograph of hyaluronic acid. Here we studied the thermal stability of hyaluronic acid. The melting point of hyaluronic acid is between 260 to 280 degrees Celsius. It shows the sharp endothermic peak and it indicates the stable presence of pure hyaluronic acid without the degradation. Second, we studied about the lambda max of hyaluronic acid. Here, the highest peak of our is 305 nanometers, and it shows the stability of UV spectrum. Then we studied the FTIR. Here, the functional groups are studied. Here, we find out the hydroxyl and carbonyl group. Our range of the vitamin C range here is 3,100 centimeter inverse, and in physical mixture, uh, it shows no reaction with the other ingredients. Then we studied the zeta potential. The range of our zeta potential is minus 31 millivolt, and it shows our formulation is stable. The negative charge is occurred because of hyaluronic acid, because in the hyaluronic acid the carboxylic group is present, and after ionization, the carboxylic acid shows the negative charge. Then we studied the particle size determination to penetrate formulation in the skin. The other value is about 187 nanometer, which is stable enough to penetrate in our skin, and absorption is better of our face serum. Then we studied the in vitro drug release. Here, the blue line represents the sample, and the red line represents the standard formulation. Both the formulation shows the sustained release over time, but our formulation shows the better drug release. Then we studied the ex vivo hemolytic activity. In this, our formulation result to be not irritating for the skin application. So that's why our conclusion is about we successfully formulated a safe and natural anti-aging serum using cow urine with hyaluronic acid and antioxidant. This novel approach offers a promising, affordable alternative for skin rejuvenation therapy.
We believe it has a great potential for further development into cosmeceutical applications. Thank you for your attention. I'll be happy to take any questions.
Thank you, Divya. If any question, ask to the Divya. Any question? Any question, Divya?
Okay, sir.
Thank you, sir.
Stop your screen.
Okay.
Next is Syed Said. Share your screen and start your
Yes,
presentation.
yes, sir. Sir, it is visible.
No, share your screen. Please share.
Okay. So it is visible.
Yes, yes. Start your presentation.
Yes, sir. So good noon to everyone. I am Sayyid Said, student of final year. My work is on fluorescence switch on off on base functionalized graphene quantum dots for highly selective and sensitive immunosensing of ferritin in iron deficiency anemia. So first we have to know what is iron deficiency anemia. It is the chronic condition in which the iron level in blood is decreases. According to WHO, there are 72% people are suffering from the iron deficiency, especially women due to pregnancy and menstruation. In the anemia, the ferritin level is decreases. The ferritin is the biomarker, which is the storage form of iron. In this work, we prepare amine functionalized graphene quantum dots by the addition of graphene oxide and 2-aminoterephthalic acid, mixed and sonicate, and then placed into the hydrothermal reaction in the Teflon line autoclave for 12 hours at 150 degrees Celsius. Then we centrifuge it and the filtrate is obtained. Then we add the antiferritin antibodies into it to we place antiferritin antibodies to increase the selectivity of ferritin. We prepare the serum which contain the components of blood and it shows the selectivity towards the ferritin. We observe that the GQDs have fluorescence and if it binds to the ferritin, it increases the fluorescence. So we add the quenching agent methyl orange. The methyl orange quench the fluorescence of antiferritin and amine functionalized GQDs and in contact with the ferritin, it regains the fluorescence. And by the measuring the fluorescence, we can detect the amount of ferritin to be present in the blood sample. We perform some analytical test and we get good satisfactory results. We perform FTIR for functional group detection. The peak showing 3,500 centimeter inverse, it is a nitrogen. And we confirm that the amine functional group is attached to it. And the XID says our sample GQDs are the crystalline form. We perform the same thing for the particle size analysis and it found to be 0.25 nanometers. We perform the elemental detection test for element detection and they found a carbon, nitrogen and oxygen. The main technology involved in our topic is the fluorescence spectroscopy. As I said earlier, the figure 7a described that the amount of quenching agent is increased, leads to decrease in the fluorescence of the graphene quantum dots. And the 8a shows that as the ferritin binds to the GQDs, it gains the fluorescence. And this method is cheaper than the existing techniques like the ELISA and RIA. In ELISA, there is antigen antibodies reaction and the special instrumented are needed. And in RIA test, there are gold nanoparticles involved in it. So the cost is automatically increases. And we use the graphene oxide. It is a cheaper one. And the aminoterephthalic acid is easily available. So we can say that it is cheaper than all the existing techniques. In last, our overall aim is that like this, we take the blood samples and we test it. So we can involve in the kit in our analysis. So to early diagnosis of the disease. And our future expert is to use the biosensor in cancer. As the ferritin level is increased in the cancer due to increase in the blood cells. So thank you. And thank you for HR Piper for providing us such a great opportunity. And if you have any questions, you can ask.
If any question, ask to the same. Raise your hand and ask them. Okay, Said, no question. Thank you. Hello, Said. Am I audible? Hello.
Yes, sir.
No question.
No question.
Next is Sneha Bari.
Am I audible, sir?
Yes, audible. Share your screen. Start your presentation.
Is it visible to you, sir? Hello?
Hello. Share your screen. Hello, Sneha.
Yes, sir.
Share your screen and start your presentation.
Is it visible?
Yes, visible. Start your presentation.
Yes, sir. Good morning, sir. I am Sneha Badi, student of B Farm. My research topic is the turning fruit peels into nutraceuticals, a natural approach to health supplement. We often see fruit peels as nothing more than a kitchen waste. But what if I told you that this is so-called waste is actually a gold mine of the therapeutic compounds with immense potential for health and medicine? Everyday homes, restaurants, and food processing plants generate an enormous amount of fruit by waste. We are literally throwing away a valuable resource that are right in front of us. And my purpose is today is to show you why we need to change our perspective. Our solution is the introduce a new paradigm, the valorization of bio-waste. This this means taking what is considered waste and extracting its inherent value. The pills are rich in antioxidants, essential vitamins, dietary fibers, and vital minerals, which is act as a nutraceuticals. These nutraceuticals compounds helps fighting chronic disease by the combating oxidative stress to boosting our immune system and supporting our health gut microbiomes. The applications are vast and scientifically approved. This global market of the nutraceuticals data represents a strong component and annual growth rate of 9.50%, that indicating a high potential market driven by the rising consumer health awareness and a shift towards a preventive healthcare. Next, this is the simplest way to prepare a nutraceutical tablets. Select the peels of the fruit, dry and grind them by the passing through the 80 number sieve. Then we perform the pre-formulation studies. Then after, weigh and powder the mixing properly and punch the tablet. These are the ingredients which are used in the formulating tablets like a banana peel powder, amla peel powder, lemon peel powder, orange peel powder, and acacia. Here the acacia act as a binder. Here the result of the pre-formulation studies of the blended powder and quality control test for the tablet that indicates the uh, tablet is effective and accurate. In conclusion, we prepare the nutraceutical tablets from the bio-waste, which is rich in antioxidant, vitamin C, fibers, and essential nutrients. They boost immunity and improve digestion and promote skin and hair health. This tablet supports overall the wellness and help combat oxidative stress. Thank you, sir.
Thank you, Sneha. If any question, ask to the Sneha.
Okay, Sneha,
Yes, sir.
am I audible? Whether you have to
Yes, sir.
perform this all practicals in your lab,
Yes, sir.
are you prepared formulation?
Yes, sir.
Okay, tell me how you will select those materials or which type of species you have to select.
Types of material or ingredients.
My question is,
Hello.
if you are selecting banana
Yes.
peel, so what type of
Yes.
species you have to select it? Means they are rich with bio ingredients or the bioflavonoids or whatever the active constituents.
Um Banana peel are rich in vitamin C or zinc, etc. We can collect the peels or foods particles that which rich is in vitamin C or zinc.
Okay, it is right, but my question is, you are from Shirpur, okay? Your
Yes.
geographical location is Shirpur. If someone is purchasing or someone trying to manufacturing those formulation, whatever you have to suggest it at south, right? So
Yes,
they need
sir.
same genus species material.
Yeah, it can be vary.
So my question is same. So species, according to the species, your active constituent will vary. Na?
I think it doesn't matter for manufacturing.
Great.
Some ingredients or vitamins may be vary, but it doesn't affect the manufacturing process
Hello, Sneha.
or efficacy.
Is it
Hello.
nutraceuticals?
Yes, sir.
So how you will be derived this? Your formulation shows the greater efficiency in human bodies. Whether performing in vitro
Because
in your studies or not.
process is ongoing. We perform only preclinical trials.
Over to you, Narendra sir.
Yes. If any question, okay, Sneha, no question.
Okay, sir. Thank you.
Mute your. Next is Patil Punam and Patil Vaishnavi. Share your screen and start your presentation. Patil Punam. I am audible, Punam?
Yes, sir.
Share your screen. Okay, Punam. Start your presentation.
So, a very good afternoon, everyone. I am Poonam Kantilal Patil, TYB Farm student from HR Patel Institute of Pharmaceutical Education and Research.
And I'm Vaishnavi Dattatray Patil from third year student from HR Patel Institute of Pharmaceutical Education and Research.
Today, we are here for presenting our study title, that is Anticycling Effect of Bacillin and Naringenin isolated from Oroxylum indicum and Citrus aurantum, an in vitro study on human sickle red blood cells. Our research focuses on the sickle cell disease, a severe hereditary disorder, and explore how natural compounds like bacillin and naringenin can potentially mitigate its effect. Now let's see the graphical abstract. So graphical abstract summarizes our study visually and it gives idea, that is the focus is on the two compounds which are derived from the medicinal plant. Now our goal was to test whether bacillin and naringenin could prevent or reduce red blood cell sickling caused by sickle cell disease, and our findings are promising. Bacillin reduced sickling by 40.61%, while naringenin reduced sickling by 37.84%, and this results suggest that plant-derived compounds could serve as an alternative for the treatment of this condition.
Let's begin with the introduction of sickle cell disease. Sickle cell disease is a hereditary disorder where RBCs lose their shape and become rigid and sickle shaped. This abnormality is due to defective hemoglobin and causes anemia, pain, and organ damage. Our study focuses on natural plant compounds with anti-sickling activity. Sickle cell anemia. The most common is hemoglobin SS or sickle cell anemia. Other types include hemoglobin SC and rare variants like hemoglobin ESD and hemoglobin SE and hemoglobin SSO, all leading to sickling of RBCs with different severity. Earlier, treatment like vanillin and phenylalanine were not effective and hydroxyurea has long-term risk. Hence, natural alternatives are being explored. In this study, we tested bacillin from Oroxylum indicum and naringenin from Citrus aurantum. The mechanism of action of sickle cell anemia is a point mutation in the beta globin gene replaces glutamic acid with valine, forming hemoglobin S. Under low oxygen conditions, hemoglobin S polymerizes and making RBCs rigid and sickle shape, which then break easily and block the Blood vessels. Symptoms of sickle cell anemia are patients present with anemia have fatigue, frequent pain crisis, and infections, complications like eye damage, leg ulcer, and stroke.
The experiment is done to analyze the anticycling activity of the compounds. First, whole blood and balance salt solution to get the appropriate erythrocytes. Compared with micromolar concentration of each compound and standard for 1 hour. They are at 37 degrees Celsius and they are incubated into the CO2 incubator. Sickling was induced by generating hypoxia, chemically using combination of 0.5% of sodium metabisulfite and 1.5 millimolar concentration of cobalt chloride hexahydrate. After that, cells immediately observed under the light microscope at the magnification of 14x. Cells were differentiated on the basis of their morphology. The cells which are concave disc-like are the normal ones, and the cells which are elongated, half-moon shape, or wrinkled are sickle cells. The percentage of the sickle cells were calculated using the formula, that is percentage sickling is equals to number of sickle cells divided by total number of counted cells into 100. The NMR analysis confirmed the structure of both compounds, bacillin and naringenin, in untreated samples. Most common underwent sickling. When treatment with bacillin and naringenin, the percentage of sickle cell was significantly reduced. Among them, bacillin shows significantly stronger anti-sickling activity compared to naringenin. Thus, both compounds demonstrated potential to protect the red blood cells from sickling. Now let's see the microscopic images. Here you can see the firstly we took the 5 samples in which A shown the normal untreated blood sample where B is treated with with sodium metabisulfite and cobalt chloride hexahydrate, sickling inducing agents. After that, C and D shows blood sample treated with bacillin and naringenin. You can notice the sickle cells compared to the compared sickling of this group is reduced as compared to other ones. This graph represent the percentage of sickle cell of different groups. Firstly, the control means untreated one group shows almost no sickling. The sickling induced group shows 100%. That is second one. That is we treated with sodium metabisulfite and cobalt chloride hexahydrate. And when we treated that sample with bacillin and naringenin, the percentage of sickle cells dropped.
In conclusion, bacillin and naringenin exhibited promising anti-sickling activity in vitro. They can be further explored as potential herbal-based therapies for sickle through in vivo and clinical studies will be needed to confirm their therapeutic role. In future, this work can be extended to animal studies and formulation development of herbal remedies against sickle cell disease. I sincerely thank my guide, our institute, and the organizer of this conference for giving us this opportunity. Thank you.
Any question asked to Puno ma'am? Vaishnavi. Hello Vaishnavi, I am audible.
Yes,
Hello?
yes, sir. Yes, sir, you are audible to us.
Which mechanism your drug has been shows anti-cyclic mechanism activity? Hello.
Yes, sir.
You have performed mechanism or not? Hello.
Our compounds are we have antioxidant properties. Oxygen yes, Hence, our reading the polymerization.
Hello.
Yes, sir.
You are taking permission from ethical committee or not while performing anti-cycling defect or anti-cycling activity.
We haven't performed any experiment. We have studied this using research articles, but it is a need for development of natural anti-cycling agents because the other synthetic have various disadvantages and toxic effects. But in
Actually,
our region, like Shirpur, uh, have been a red zone for sickle cell anemia. So we have taken this topic as a need.
naringenin is standard or polyphenol. Hello.
Sorry, sir. I haven't
Naringenin,
listened.
naringenin is polyphenol or flavonoid.
Sir, it is flavonoid. It
What
is
do you say?
usually obtained from the papaya fruit.
You have extracted or directly procured?
काय बोलताय ते?
What do you say?
What, sir, we are not able to hear what you are saying.
You have extracted or directly procured?
Sir, we directly get it compound. We didn't extracted it.
We didn't extracted it, sir. We have studied using research articles.
Okay. What do you mean?
Okay, tell me, Poonam and Vishnavi. You
Yes,
have
sir.
taken all the data from the research manuscript. Okay. So my question is, do naringenin and biacidin modulate hemoglobin polymerization?
Sir, we didn't get the question. Can you please simplify it?
Can naringenin and biacidin modulate hemoglobin polymerization?
Sir, we didn't have any idea about that.
Thank you.
Hello, Poonam.
Yes, sir.
No questions. Thank you.
Yes,
Stop your
sir.
sharing. Next is Gargi Patil and Roshan Patil. Share your screen and start your presentation. Patil Gargi,
Yes, sir.
share your screen.
Sir, are you visible? Visible our poster?
Yes. Start your presentation.
Good afternoon, everyone. I am Roshan Satish Patil and my co-partner Kagi Pramod Patil from Asian Paper Shirpur. Our research interests are in veterinary science, artificial intelligence, and animal welfare. Today we are happy to present our poster on advancements on artificial intelligence technology for improving animal welfare, a veterinary clinical practice and biomedical research. The development of AI in recent years has changed the focus of biomedical research, particularly in the highly promising field of translational research This study explored the transformative impact of AI on livestock industry. Animals play a crucial role in our ecosystem and coexist with us, forming so AI can the and in the and can also help in better feeding so animals get right nutrition. Sir, can you audible? Hello, sir, can I audible?
Yes, yes.
Basically, in healthcare, AI tools can detect diseases early and guide treatment, making veterinary care more proactively. In and mainly in research, AI support work on the cancer, drug and vaccine development, antibiotic resistant genetic diseases tracking, and more. This make it an important tool for solving the complex problems in animal health. For example, precision level farming, which helps to reduce the stress. Stress is simultaneously affect on the microbial populations and fermentation process in animal human, which is decrease the feed intake in animals. Various roles of AI in veterinary science, as like animal health maintenance, by using the various sensors. AI monitor vital parameters such as heart rate, body temperature, and activity levels, like animals, enabling early detection of health issues. Environment and AI also useful in environment monitoring of the environment in animal surrounding, as like the temperature, by controlling the temperature, humidity, providing insight to maintain the optimal living conditions for animals. It is also help in diagnosis purposes by using the analyzing radiographs, ultrasounds, MRI, and CT scan, fast and accurate diagnosis of fractures, tumors. It is also helpful in the pathology and cytology. AI system analyze blood, urine, and tissue samples, cancerous cells. We can also identify the various works or various reactions of animals, such as by using smartphones as an ECG device for dogs at home. Uh, we can also use ear tag sensors, which observe the diet, temperature, behavior, mobility of the animals, and track vital indicators to look for early sickness symptoms. And also the microphones, which are used to track and detect the audible symptoms of diseases. Uh, we can also use the sensors like electric conductivity and somatic cell count, which is used to recover the mastitis, which basically occur in cows. AI also helps to optimize the vaccination schedule by analyzing herd immunity levels and individual animal health data. It is also play a crucial role in management of the nutrition in animals, as the system recommended dietary adjustment tailored to individual animal to boost immunity and prevent the deficiencies. It is also used to recover the customized treatment protocol based on an animal's specific health data, reducing recovery time and improving the outcomes. It is also analyze large data sets from drones, cameras, and sensors to monitor herd health, optimize grazing patterns, and improve productivity. It is also helpful to monitor milk composition and suggest improvement in feeding and care to enhance milk production. This sensor detects mastitis in cows by analyzing milk quality parameters as like the electrical conductivity or somatic cell count. It is also helpful to training students, which is further study in research in animal welfare, and also discover of new veterinary drugs and vaccines by
simulating biological process and analyzing large databases. In conclusion, AI utilization of AI offers sustainable potential for enhancing animal welfare, and it is capitalizing on advantages, addressing limitations and concerns, and exploring the future direction. So it is very essential for diagnosis the every diseases or any kind of problems occurring when in animals, and to address it is essential to continue research in responsibility for interdisciplinary collaboration. Thank you for giving. Thank you. Thank you for giving your time. Happy to take it for
Gargi, am I audible? Hello. Hello.
Hello.
Gargi, am I audible? Hello.
Yes, sir.
Can you elaborate? AI is used in biomedical research. Which biomedical research?
Hello.
Hello, am I audible?
Sir, could you speak loudly, please?
How AI is effective in biomedical
Yes.
research? How AI is effective in biomedical research and in which biomedical research? Can you elaborate?
Sir, it is used as a for preparing the vaccinations and by diagnosing the various kind of diseases in which I don't idea about specifically which kind of biomedical data research, but it is used to identify the various kind of diseases, and by using various sensors and microphones, we can analyze the more all information about the animals.
And AI used in animal welfare.
Yes, sir.
In which this is the diagnosis we are used.
We can diagnose the mastitis in cows by using the electric conductivity sensors and somatic cell count sensors, and simultaneously we can also diagnose the blood influenza virus which cause in birds.
If any questions?
What role does AI play in veterinary epidemiology? Yes, Gargi.
Hello. Yes, sir.
What role does AI play in veterinary epidemiology and outbreak prediction?
Sir, please speak loudly.
What role does AI play in veterinary epidemiology? You know, previously I think 2 to 3 posters before lumpy skin disease is presented by the Divya Thakres. So it is useful in those conditions? AI.
Yes, sir. AI is used in every type of disease in causes in animals because
How
uh,
can AI
we
help in
prepare
optimizing?
by by using the sensors. We can put the sensors in animals in specific organs as like their legs, as accelerometers in ear temperature, which identify the temperature, pH, and also we can set the
हाँ
sensor in
सर.
their torso sections, and
Ethical
also in
concern
नहीं सर
is
चालू केलं
important.
पोस्टर
saliva hair.
ते 2 जणांचं व्याख्या झालं तुमच्या DVRचं ते चालू आहे.
Sorry, sir.
ते कॉन्फरन्सचं
I
पोस्टर
can
प्रेझेंटेशन
not
चालू आहे.
audible.
मी पाठ ते ऑनलाईन आहे सर ते लॅबला बसलेले आहेत मी पाठवतो त्यांना झाला का
नाही दाढी त्यांना
मग मी
फोन
पाठवतो
करते.
सर घेऊन घ्या.
नेक्स्ट स्टॉप.
मग मी घेऊन घेईन ना सर मी तुमच्या समोर. हा सर मी चालेल मी पाठवतो त्यांचं झालं का नाही. ओके थँक्यू गार.
थँक्यू.
Now,
हॅलो.
thank you. Piyush. Hello, Piyush. Hello. Patil Piyush.
Hello, am I audible, sir?
Yes. Share your presentation. Okay, start your presentation.
So we can see the post. Hello. We can see the poster, sir. Hello. Hello. Visible, sir? Yes, sir. Hello, everyone. Good afternoon, sir. Today, I want to talk about the exciting new technology that could help solve the global water storage. A hydrogen embedded membrane of atmospheric water harvesting. This is the novel and eco-friendly approach to the collecting the fresh water from the direct atmosphere. The globally many regions face water scarcity. Efficient system that takes this the by creating highly efficient system that capture water vapor from the atmosphere. Our technology uses the key components metal organic framework or MOS and hydrogels. MOS are specially material with the high porosity and large surface area, which makes them the excellent and absorption water vapors from the atmosphere. Hydrogels. Hydrogels are the intended to the enhance the water retention. They can absorb a sufficient amount of the water and then release under the specific condition. The most important part of our design is the vertical aligned nanosheet structure. This arrangement improves the efficiency of the water vapor absorption, helps the water flow in the right direction, and makes it the condensation process more effective. This design helps our overcome the limitation of the single component MOF absorption, which have the poor water retention. Our system works by the two-step process: water capture and water release. This vertical aligned nanosheet capture water vapor from the direct air, and water release is the capture water is condensed and released from the hydrogel, offering a low energy solar driving method to create the fresh water. Our system shows the very fast water absorption, reaching 9.4% saturation in just 15 minutes. It also maintains its excellent performance when even after the reduced cycle. The material are used in this project: chitosan and PEG, polyethylene glycol, zinc and zinc nitrate. The shows the temperature graph, temperature table. Hello. Hello. Yes. This temperature table shows that the CTPV is the temperature of the material start the 24 degrees Celsius and gradually increase the 37 degrees Celsius after the 600 seconds. After the HMO and CTPV, this material start the 24.2 degrees Celsius and reaches the 48.0 after 600 seconds, showing a much faster higher temperature increase the capture to CTPV. Last is the MOF and CTPV. This material shows the most significant temperature increase the starting at 25.4 degrees Celsius and reaching the 59 degrees Celsius in the corresponding thermal image through the table value of the 600 seconds is not fully visible. The, tab, uh, the table and thermal image demonstrate that the material containing MOF nanosheet HML CTPV and MOF CTPV heat up much more efficiently than the hydrogen membrane alone. At the conclusion, the metal organic framework harvest from the atmosphere, water harvest from the atmosphere. This system is the high efficient with the fast water absorption rate 9.4% saturation in just 15 minutes. It also has the low energy demand and excellent cycling stability, making it from a promising solution for the providing sustainable water source in dry regions. Thank you. If you have any question, ma'am. Yes, ma'am. Yes, ma'am. Ma'am, metal organic is hello. Nano sheet. Hello. Yes, ma'am. We are use the solar panels, ma'am. Hello. Just like uh PVA polyvinyl alcohol, ma'am. Yes, ma'am. Ma'am, hydrogen membrane is sustainable made from the chitosan and PVA. Enhance the water retention, ma'am. Enhance the water retention. The capacity. Enhance the capacity water retention. Thank you, ma'am.
Okay, Piyush. Ma'am, conclude this poster presentation.
Absolutely.
Hello, ma'am. Overall is the poster presentation. Okay. Yes, ma'am. Yes, ma'am. Sure. Thank you. Yeah. Thank you. Thank you. Okay, thank you. Yes, yes, yes.